Yes, doctor.
Yes,
Yes,
doctor.
doctor.
You can
Yes,
hear me,
doctor.
okay? Okay, okay. How's everyone? Everybody's okay?
Okay, doctor.
Good, doctor.
Good,
Okay,
doctor.
Fine, doctor.
okay. Have you had breakfast?
Yes.
Okay, yes,
Yes,
already.
doctor.
Okay,
Yes.
okay,
Good,
that's
doctor.
good. Please make sure that you have breakfast before you attend my class. In fact, you should have breakfast before you attend any classes. Sometimes you may not have, well, at the moment, most of you at home. Sometimes you may not have enough time if you're rushing, but then again, if you wake up at 6 o'clock or 6:30 in the morning, you should have ample time to have breakfast. Please have breakfast because it's a very important meal. Even if you cannot have a big breakfast, just grab maybe bread, or make a sandwich and maybe a hot cup of Milo. But it's important to have a good breakfast before you start classes. I always make sure last semester or previous semesters when my classes were at 8 o'clock that students must have breakfast before they attend my classes. So now the department has changed it to 9 o'clock. I still want to make sure that students have breakfast. Okay, right. So before we start the lecture, a few housekeeping matters. Number one, we're going to use the same link for lectures on Monday and Wednesday, which is at 9 a.m. Um, this should also be available on Spectrum. For Tuesday, you have another link that will also be available on Spectrum. But we only start Tuesday classes next week. Now, normally my Tuesday classes always start at the second half of the semester, meaning for the last 7 weeks. But because this semester I'm going to do some changes because of the assessment directive that we have. We're going to have to change some of the assessment method, especially for final year examination. So I'm going to move it slightly earlier, meaning that you're going to have your Tuesday classes starting next week for the next 7 or 8 weeks. Okay, and then after that you're going to be free after mid semester break, one or two uh, weeks after semester break. You're going to have free time on Tuesday. So I don't use full Tuesday classes for the whole semester. I only use half. That is in your student learning time, how we calculate it, and it's only half. But if there's a need arises, um, for example, we have classes that need to be rescheduled or classes that need to be replaced, then I will use the Tuesday slot, only the free Tuesday slot. Okay, so I repeat, you can use the same link for morning lectures on Monday and Wednesday, and there's another link for Tuesdays only. Well, Tuesday classes only starts next week for the next 7 weeks or so. Okay, so that is the first announcement. The second announcement, within this week or by the end of this week, I will post on Spectrum um, the schedule for all your all your topics that we covered, because we're going to do it week per week, and we're going to have that schedule week per week. And then I will also detail the assessment type. Remember last time I mentioned that we have 50% for continuous assessment and 50% for final year examination. The 50% for continuous assessment part will not change, okay? But the 50% for final year examination will change, because for the last few semesters when we do this online teaching and learning, I run an open book exam during exam week. But this semester, because we have a few more changes, and I'm going to make some changes on that part. Okay, but you will know by the end of this week, hopefully, if not earlier, so that you can plan, and then you can rest your mind. Otherwise, you keep on thinking how the assessment is going to be made. For the continuous assessment, now for the continuous assessment is important. The 50% that composed of assignments. You'll be given assignments, and don't worry, you'll be given ample time to complete the assignment. And you will also have pop quizzes. Now, these pop quizzes, normally during the face-to-face time, we do pop quizzes, meaning that when you enter class, suddenly you end up having to spend 10 minutes to answer pop quizzes, which is on paper. But when we start this online, I changed the pop quizzes to what I call the clicker quiz. Now, the way the clicker quiz works is that For 10 minutes or 5 minutes or so, depending on the length of the quiz, during the lecture time, it could be either Monday or Wednesday or even Tuesday, and these are unannounced, meaning that it's never announced, it just pop up at times, so it's a surprise. So you'll be given a clicker quiz. Now the clicker quiz works that I will then portray the sets of questions. You will get ready with a set of what we call the answer sheet. Don't worry, that will be available on Spectrum as well. And then the clicker quiz works that for 1 or 2 minutes, depending on the length of the question will be displayed on the screen, and you have to pick the most accurate answer based on the question that's been displayed on the screen. Okay, so that's what we call clicker quiz, meaning that I will click, you will answer, I will click, you will answer, and that will go for 5 or 6 minutes or even 10 minutes, depending on the number of questions. So you're going to have quite a number of pop quizzes throughout the semester. That number will be detailed later on, but just be prepared because it's unannounced. Sometimes it can be at the beginning of a lecture, sometimes it can be at the middle of a lecture, or sometimes it can be at the end of the lecture. And it's unannounced because it could be on Wednesday, or it could be on Monday, or it could not be on any that particular week. So the reason being is that I do not want you to study last minute, number one. Number two, I want to make sure that you are always on your toes when it comes to revising your studies. And number three, because genetics involves a lot of concepts rather than memorizing. And I mentioned last time that you cannot remember or appreciate what you do not understand. So you must understand, and in order to understand, you must read and revise, and only then you can get the concept right, and only then you can answer questions right. This is to make sure that you've been trained and build up your comprehension over time, meaning that you're not allowed to cram. Okay, and we really want to make good use of the 14 weeks available to us. Now, don't worry, before we start the clicker quiz, I will do a trial run. Now, the trial run is mock, meaning that it's not marked, so don't worry. We just want to do the trial run so that you are familiar with what clicker quiz is all about. Okay? Any questions up to that point?
Doctor,
Yes?
what if we absent during the quiz?
Sorry, Faiz, I can't hear you. Can you repeat the question again?
What if we absent? We didn't attend the class because of some personal issues during the quiz.
What is the absence?
What if?
What if? Oh, what if when you attend the class? Okay. What if when you did not attend the class and then suddenly there's a pop quiz or clicker quiz? Is that the question?
Yes, doctor.
Okay. So normally, when students did not attend classes and then there's a pop quiz, if the student cannot provide evidence of why he or she cannot make it to the class, okay, then he or she will forfeit that clicker quiz marks. If he or she or the student involved can present evidence, for example, a medical certificate or an official letter, or depends on the circumstances, then another clicker quiz will be arranged at the end of the semester or before the end of the semester, depending on the schedule. But I would very much suggest that do not miss the clicker quiz, because when you have to take the clicker quiz at the second section or at another rearranged time, students sometimes find it overwhelming because at the end of the semester they have so many things to do. And there were in the past students who missed up to 3 clicker quizzes, and he or she had to sit for 3 subsequent clicker quizzes in one sitting. And I know that can be a bit taxing on the student. So the moral of the story is that do not miss classes if you can. But if you cannot and you have good reasons for it and you have evidence to support it, then another session will be arranged for you later. Okay?
Okay, thank you, Doctor.
Okay, any more questions? Okay, so if that is the case, don't worry, as I said, by the end of this week, if not earlier, I will put up materials on Spectrum. And normally when I put materials on Spectrum, I will also put up a message for you saying that, okay, there's a new materials on Spectrum, you can come and check and so on and so forth. But I also know many of you check Spectrum periodically because I can check who has been logging into Spectrum and check what's in for the courses. The system allows me to check even the date and when the person or the student checked, so I know. Okay, and so far I noticed that some students who never even access Spectrum for the course. So it's okay, it's still the beginning of the semester. But as I said last time, it's your second year, you really want to make good use of the time. Okay? Everybody okay?
Okay, Doctor.
Yes,
Okay,
okay, Doctor.
Okay,
Okay,
Okay. Okay. So now we're going to start with our lecture. Oh. Sometimes the computer tries to be smarter than I am. Wait. Right. Can you see the screen?
Yes, Doctor.
Yes,
Okay.
Doctor.
We're going to start with our lecture. Now, the first one is, well, I call it lecture 2 because we already done the introduction. Now, this picture is from Welcome Image Award. This was a while ago, but I like this picture because it shows the sequence of cell cycle in one particular picture. Okay, the sequence of cell cycle in one particular picture. Now, in this picture, they use fluorescent microscopy, and they actually tag or probe the cell membrane in the fluorescent color of blue-green here, and the genetic material, which you can see clearly as a set of chromosome here in red. Okay, so it's a nice picture because it shows the sequence of a cell division and cell cycle from only one part, and then it moves, and then you have this segregation and movements of chromosome into two parts, two poles, and then it's segregated, then completely starts of cytokinesis, and then you have a division. So it's a very nice picture showing the sequential part of the cell cycle from one cell, and you give two daughter cells. Okay, so it's a nice picture. So from here you can see that cell cycle and genetics per se is all about growth, development, propagation. Okay, so you have a single cell, now you have two daughter cells, and each of these daughter cells can undergo either mitosis or meiosis to produce more cells, depending on what you need. So mitosis, remember, you produce identical cells, identical daughter cells, whereas meiosis, which is for gamete formation. So when you have gamete formation and you have meiosis, which come in two stages, it is to produce only half of that, because in any organism, when fertilization occurs, the size of the genome then will be complete, one from the mother and one from the father. Okay, so this is a very nice picture. Now this is another picture to show you the same, but unlike the previous one, it does not tag or probe the cellular membrane, but it shows a much clearer way of how chromosome being packaged and then how they're being segregated into two daughter cells. Okay, now you have here, which is an interphase stage, where you can only see the chromosome as chromatin, which is not highly condensed. And then you have condensation of the genetic material into what we can see clearly as chromosome. Okay, here, much more condensed. And then allow for the division of the chromosome, or karyokinesis, if you may recall when you're in high school, karyokinesis, and you end up with two daughter cells, again in interphase stage. So one might want to ask a question, why must a cell divide? Anybody would like to try? Why must a cell divide?
To replace the dead cells.
Sorry?
To replace the dead cells.
That's one example, to replace a dead cell. Any other functions? Any other reasons? For growth? Okay, for growth. But there was an interference just now by ISAP for growth. Any other else? For now, repair, for growth, anything else?
For unicellular organism, cell division produce new organism.
That's reproduction for unicellular organism, yes. Anything else? Okay, all the answers are correct. Basically, a cell divide to perform various functions, for growth, for repair, for further development, for gamete production, and of course for unicellular organism, binary fission, i.e. to produce a new individual. Okay, so basically you just want, if we take into mitosis, basically you want to produce two daughter cells. So it's like photocopying machine. You put a piece of paper containing your lecture notes, and you expect the photocopying machine to copy that lecture notes exactly, so that you end up with two. So the whole system is like photocopying machine. You want to make sure that the genetic material produced when you copy that is the same, because you are not going to be a happy person if you put your lecture note into a photocopying machine and that photocopy machine photocopy into something else. Okay, so just remember, the analogy is very simple. When we have cell cycle and when we have genetic information being passed from one generation to another generation, is to make sure that the fidelity or the correctness of the genetic information passed from one cell to another cell is ensured, meaning that there's no changes. For mitosis, yes, and even for meiosis, because for meiosis, of course you produce only haploid, if you only half, but if you have genetic information being wrong when you have meiosis, you're going to have a problem as well. So just remember, genetics is all about blueprint. Why we need that blueprint is to perform various functions of life. Okay, you need a plan, you need a recipe. And when you need to have used that recipe, every time you put or copy the recipe, it might be the same recipe. If you have got wrong recipe, you're going to get wrong food, everybody's not going to be happy. So just remember, genetic is all about copying that information so that that information is correct. If that information is not correct, if you have a mitosis, for example, you're going to get diseases. Cancer, for example, is genetic disease, because when the genetic material, the blueprint, just go all over the place. Okay, and then you can also have genetic diseases if, for example, the gamete carries information which is not correct or carries some sort of mutation in the genetic material. That can affect the outcome. Okay, so now just remember, the analogy is like you want to have this copied to produce two identical copies. Okay, just remember, that is the simplest analogy I can give you. Photocopying machine. Just want to make sure that the photocopying machine is working well and is working correctly, so that whatever you put in the copy machine will be copied accurately. Okay, are we all on the same boat now?
Yes, Doctor.
Yes,
Okay.
Doctor.
Okay.
Yes.
Now, we mentioned this before. Now, the ability of organisms to reproduce best distinguishes living things from non-living matter. Viruses is an exception, okay, because they hijack cellular machinery in order to produce more copies of themselves. Now, the continuity of life is based upon reproduction of cells or cellular division, and cell division is an integral part of cell cycle. Okay, I mentioned this before. Now, the cycle begins with replication of genetic material, in this case your DNA, followed by cell division to produce two daughter cells from one parent cell. So that's the copying machine. Must make sure that the genetic material is copied correctly from the parent cell to two daughter cells. Okay. Now I mentioned before that the machine must be working properly. So that's why cell cycle is a highly ordered process and is a tightly controlled step by the cell cycle machinery. We'll see later on what the cell cycle machinery is all about. And if you want to know more about the cell cycle machinery and how they control it, there's an elective course for your third year. Okay, so now you've encountered this before, but let's just go through this again. There are different types of cellular division. You know that we have prokaryotes, which is binary, via binary fission. This is primarily for their asexual reproduction. But for eukaryotes or eukaryotic cell, we have mitosis and meiosis. And why mitosis is needed? Because it's important for growth, it's important for development and repair. And it involves somatic cells, and they produces only genetically identical cells. So it's 100% copying machine. Whereas meiosis only happens in specific reproductive cells. Okay, and they produce genetically different cells but carries only half the number of the chromosome of the parent cell. So every time you produce only half. Why? Because half from male and half from females combined produce a complete whole. Okay, so that's why you only produce half of it. So gametes, either eggs or sperms, or in certain organisms like ferns, produce spores, are involved in sexual reproduction. Okay, so that's why you have to make sure that the cell cycle is tightly controlled. Why it must be tightly controlled? Because the genetic material must be transferred correctly. I repeat this many times. Why you need to control that process? Because you want to make sure that the genetic material is transferred correctly. It's just like, not just like copying, photocopying machine, or even just like, when you order food from Grab or Foodpanda. If that process is not controlled, you order, say, nasi lemak, and if that is not controlled, the rider might got it wrong and deliver you something else. How do you feel? I don't think you're going to be happy, Panda, because you got something different than you ordered. So control process is very important. Okay. Now, you've seen this before. This is natural structure of cells labeled A, B, C, D, and E. So what is K? Anybody? What is K?
Centriole.
Yes, okay, centriole. What is A?
Mitochondria.
Partly correct. Mitochondrion, because we refer to only one. Mitochondrion is one, and mitochondria is plural. Okay, so remember, in biology, singular and plural is important. So that's partly good. How about, let's see this clearly. How about, there you are. What is that? H. That thing. Hey, come on. Your second year already, you should know that within seconds.
Ribosome.
Nucleus pore.
Apa dia? Not
Nucleus
ribosome.
pore.
Ribosome is L. What is that?
Nucleus pore.
Nuclear pore, yes. Okay. So that's good. So if you're not sure, make sure that you brush your cytology again for ultrastructure of the cell. Okay. Now, cell division, process by which a cell, including the nucleus, undergoes replication and split to produce the daughter cell. Now, when we talk about cell cycle, the most important one is the nucleus division. It's the nucleus, because that's where the genetic material is residing. So in order to be viable or living, each daughter cell must contain a complete set of genetic material so that all its proteins can be expressed at appropriate levels. Now, this is important because complete set of genetic material is like having the same recipe book from your mother. So when you photocopy that recipe book, you want to make sure pages 1 to 10 is correctly the same. Why? Because, as I mentioned last time, genetic material, your DNA, is a blueprint of life. So your protein or everything being produced from that blueprint of life must be expressed at the appropriate level. If you got it wrong, we will see later on where you either, one, you cannot produce the protein or gene product at all. 2, you produce less than what you needed. 3, you produce too much of it. 4, you produce it at the wrong time in the wrong place. All that can give problems, either diseases or abnormal cellular function, tissue function. So we need to make sure that it is expressed at appropriate levels. Now, in my last lecture last time, I said that everything in life, especially in your cellular or system, is very dynamic, because biology is very dynamic. It's very dynamic. So things change quickly over time, very quickly, when and where it's needed. So that's why it's important for it to be expressed at appropriate levels. If your body requires hormone A at 50 micromolar in your blood vessel, you cannot produce less, because it will not be able to be as effective as you want. If you need an enzyme C, for example, at 1.5 micromolar, okay, you need to have that level, otherwise it won't function properly. So that's why having the materials expressed, or the gene product or the protein being expressed at the correct concentration in your body at the correct time is important. And that relies on the genetic material, or your DNA, on the blueprint of life. So basically, if the recipe book is wrong, the outcome is wrong. If the software is wrong, the hardware will not function. You must remember that. Okay, in genetics, that's what we appreciate. Why is important? Because it's blueprint of life. Everything about life depends on having correct information. Okay. Now, so in addition to its role in directing protein synthesis, DNA also enables the passage of genetic information from one generation to the next, which is for reproduction. Okay, so in sexually reproducing multicellular organism, cell must have two mechanisms of cell division, resulting in both diploid and haploid daughter cells. Diploid is in mitosis, and haploid in meiosis. Okay. Now, eukaryotic cell division composed of mitosis, the division of the nucleus, and cytokinesis, the division of cytoplasm. What is the name for division of chromosome? I mentioned it during the first slide. Now, what is the name for the division of chromosome? Somebody, please say something.
Karyokinesis.
Correct, karyokinesis. So, don't mix things up. Mitosis is the division of the nucleus. Karyokinesis is what happens when you divide the chromosome. And cytokinesis is what happens when everything is completed. That mitosis is completed, karyokinesis is completed, and cytokinesis is the end of it. Meaning that you have a division of the cytoplasm. Once your genetic material has been completely duplicated and ready to be separated into two daughter cells. Okay? Okay. So cells duplicate their genetic material before they divide. Each daughter cell receives an exact copy of the genetic material DNA. So a dividing cell duplicates its DNA and allocate the two copies to opposite ends of the cell, and only then split into daughter cell cytokinesis. So it goes in sequence. So you have karyokinesis, mitosis, cytokinesis. Everything is in order. Okay. Now it's the same thing. Everything in life follows order. Everything in biology requires organization, and everything is programmed. Things does not happen by chance. Okay, so everything is programmed. That's why biology is the most beautiful system, because it's tightly controlled, but it's also very dynamic. Okay, So this is the structure of the cells that we talked just now. Now, I'm going to talk about the organization of the chromosome. Why is it packaged as such, and why it is important to have it be packaged like that? Okay, so in my first lecture, I showed about Roger Kornberg, and Roger Kornberg is the one who contributed a lot on how we understand the packaging and the expression of eukaryotic chromosome. Okay, so let's start here. Okay, before we start there, I just want to have to you this one particular picture. Okay, now, when you have these pictures in interface, okay, you can't see the chromosome very clearly, but here you can see them very clearly, correct? You can see the chromosome very clearly. Now, the question is, why can't we just use the chromatin and just divide them? Who can answer that? Why can't we just let the chromatin swim freely and divide into two daughter cells rather than having it packaged into chromosome? Anybody? The question is, why you can't allow genetic material in chromatin form, which is highly diffuse, to just being transferred into two daughter cells without having this kind of packaging into separate chromosome? Hello?
Something might gone wrong during the mitosis because there's so much chromatin. They are not tightly packed.
Okay. That is a good direction. Let's refine your answer. What's your name?
Yap Sun Yao.
So I call you Yap. Is that okay?
Okay.
Okay. So that is going towards the right answer. Yeah. So can you refine that answer? You said that something might go missing if it's not packaged. Can you refine it further? Go on. You're on the right track, but I just want to see a refined answer. I just want to hear a refined answer.
I don't know.
Okay.
I
So
have no
basically,
idea.
okay. It's okay. That's a good attempt. Yeah. Basically, what Yap said is correct. You want to make sure that you do not want to have it in interface or in chromatin stage because it's a diffuse. It's like spaghetti. So when you want to transfer spaghetti from one plate to another, if you do not package it properly, you might lose some. One plate might get more spaghetti, one plate might get less. One plate might get the meatballs, one plate might not get the meatballs. Even the number of meatballs might be wrong. So why you have to package it properly? Because you are dividing into two daughter cells. You are dividing into two groups or two separate cells. So you want to make sure that it is packaged properly. It's the same thing when you try to move houses, or you want to move from house or your village or whichever. I do not believe that if somebody would just say, okay, I need to move house, I'm just going to take everything and put in the car, and then when I get into the new place, I'm just going to unload everything into the, because chances are you might use something. Okay, so that's why when we move houses, when we move between villages or whatever it is, you package everything inside a box or boxes or bags so that you don't lose stuff. Okay, so that's why in biology it's the same thing. You want to make sure that everything is packaged like this so that it's easier to move things around. You reduce any risk of missing things, and you reduce the chances of anything wrong can happen because everything is packaged properly, and you can just move the package and make sure that you have 23 packages, 46 boxes. All 46 boxes have been transferred correctly. Okay, so that's good. Okay, so the same thing with your bacteria. We will always say, oh yes, double helix and so on, but they really appreciate what double helix is all about. So we know here is a double helix. We know this is, and this is where genetic materials is composed. You have CGAT, and of course you have genes, which is a sequence of the DNA. And but these are two of them again, okay, complexly twirled here, and then it wraps around a nucleosome core of 8 histone molecules. So here is the 8 histone proteins. Okay, there's the 8 components there. It's an octamer. An octamer is 8 histone molecules. Okay, I'm not going to name them, but you can check them. But for the purpose of this course, you just know that there are 8 of them. And here you have what we call the H1. This is the linker histone. Okay, the linker histone, H1. Now, what it does is that when you have this wrap, you have this wrap around the histone core. Okay, and the histone H1 acts as a stapler, making sure that what you wrap around the histone core stays in place. That is important because if you don't staple this in place, you can't move and they will not stick to the histone core. And that histone core is important because it wraps like around, when you eat spaghetti, sometimes it's difficult to move the spaghetti around, but if you wrap the spaghetti around the meatball and you just need to move the meatball, the spaghetti will move with it. Okay, so imagine that this is a meatball and you have a spaghetti wrapped around the meatball. When you want to eat the spaghetti, you just move the meatball, the spaghetti will move with the meatball. So it's the same thing here. Sometimes people call it the beat on a string. Why? Because beats and string. I prefer spaghetti and meatball because students respond better when it comes to food. So here, it wraps around and you have the histone H1. And this allows us to have furthermore folding up and coiling. And this is then coiled even further into these loops and even further. And if you have all that, that's why you end up with the chromosome because of this light coiling. And this is what you can see under a microscope because this is visible. This you can't see under the microscope. No way you can see this. This you can see because it's tightly condensed, highly condensed, tightly coiled, and this is what you can see. Okay, so when it's in chromatin form, you can't see this way. It's only when it's in mitotic stage, i.e. as a chromosome in mitotic stage, is tightly packaged like this. So when the cell is in the interphase stage, it's very active because the transcription and translation actors need to access the DNA materials in order for it to produce gene products. But when you need to do this, it is high-techly packaged that you don't need to have it access as per se at any of this stage because you are packaging to move things around. Okay, you get that? So now when we look at this same thing, you have the DNA double helix, and you have the histone, and you have the histone H1, and you have the nucleosome bead, and this is what they call beads on the string, or I call it, spaghetti and meatballs, and you can see this like this. And if you have this tightly coiling nucleosomes all packed up like this, okay, 30 nanometers wide, you can see this clearly. But it is 30 nanometers wide, it's only a small fraction of this, which is riding on a protein scaffold. Okay, and now that increased by 10 times, 300 nanometers in diameter. So that rides on the protein scaffold, you can see that. And then that protein scaffold is then wrapped around further, and when you wrap that, it's only a small fraction of that. So can you see now how your DNA is now packed into this chromosome in order for us to move things around? And you can see it clearly under the microscope if you stain it properly. Right? Of course, we see different staining methods later on when we look at the karyotype. So why does we decide to organize in such a manner? Yeah, already answered that. Because we want to package this properly and we want to make sure there's no problem or issues regarding the correctness of the information, or fidelity of the information, security of the information being transferred from one cell to another. Okay? And we still at the organization of chromosomes. So the chromosomes are all about DNA. We also know that much of the structure consists of various proteins. I showed you just now. So don't mix these up. Chromatin is one of the two identical copies of DNA making up a duplicated chromosome and they join by the centromeres. Sister chromatids form for replication. Okay? Through replication, remember, you have the duplication of genetic material, so you duplicate the chromatin. Sister chromatin are basically the same. Okay? And then chromatid remains as chromatid as long as it's held in association with the chromatid at the centromere. So here you have one chromosome duplication into the DNA synthesis, and you have the sister chromatids because they are basically copies of each other, and separation of sister chromatid into different cells. Okay, so remember one duplicate and it transfers to two, and of course either is mitosis or meiosis is always segregated into two daughter cells. Okay. Now just a repeat. So this is the graphical chromosome, centromere, and chromatid. Okay, so a chromatid remains as a chromatid as long as it has association with a chromatid at the centromere, which is complex, a huge complex of protein, and in fact, it's the one of the most studied complex of protein. You can even do a PhD by just looking at this. Okay, because it involves a lot of proteins, a lot of signaling, a lot of pathways, and people are interested in studying this because if you have something wrong with this, you're going to have problems segregating these two. So that's why people study this a lot. Okay. So when two sister chromatids separate after metaphase, they are referred to as sister chromosomes. Because why? It's like this. And duplicated. And then segregated. Okay. So same here, you have the homologous chromosome and you have the duplication, you call it sister chromatid, and you have the separation into two daughter cells. Now, these are homologous chromosomes, sister chromatids, but one of these is not the same as the other. They just duplicate and replicate and duplicate it into the cells. Now, karyotyping. Karyotyping 1, 2, 3, we already know. The reason why we have karyotyping, this one is the most classic type of karyotyping. We use gene cells, and that's why you get the black and white bands. Those bands are used to categorize the chromosome. So that's why we can classify them into 1, into 22, and of course you have the sex chromosome. Okay, so in this case the G bands or karyotype for human female, and of course you can also have for human male. Okay, so that's it. So when we go back to chromatid versus chromosome, it's the same story. You just need to know which one is being referred to at what stage. Okay, so again, I repeat this many times because sometimes, every year when students answer either the assignment or especially the exams, they tend to get things mixed up. That should not happen for second year students who always get things mixed up because you should know which one is which. Okay, so chromosome, chromatid, so they are there. So chromatid is unknown DNA found throughout the interface. They are unknown because DNA is being utilized in the synthesis of various macromolecules and gene products. It's like a book. In order for you to read the book, you must open the book. Okay, but when you want to transfer the book from one place to another, you close the book back in the box. So chromosomes are tightly packaged DNA found only during cell division, and DNA is not needed in macromolecule synthesis but replicated for replication and transport. Okay, that's the next stage. Okay, so cell cycle, we already know this. Okay, daughter cells, cell growth, chromosome replication, chromosome segregation, and cell division. So this is the cell cycle. Now, duplication of cellular contents, followed by cytokinesis, producing two genetically identical daughter cells. This process, the cell cycle, is a one-way street. It cannot reverse. I repeat, it's a one-way street. It can only happen in this direction. It cannot reverse. Okay, and that's why it's important why the cell cycle is tightly controlled, because you cannot reverse. Once it goes, it goes. Okay, but there are mechanisms in place. To make sure that everything is in order, everything is correct. I said that it's a one-way street, it's a one-way process. It cannot be reversed, but it can be halted, meaning that it can be stopped. If there's something wrong, it will be stopped. Say, for example, you have a mutation. If it's detected, it will stop the process and try to repair. That's why we have the DNA repair mechanism. Once it's repaired, you can move again. And then suddenly you have missing chromosome, you stop. Once the missing chromosome is relocated, then it can go again. So that's why, like I said, it's a tightly controlled process. And there's a lot of signaling, a lot of proteins, a lot of actors and factors going on to make sure that this happens. So once that happens, human being is about 22, 24 hours. So you can imagine before that process being completed, there's a lot of process going on. You don't know anyway, you don't know what's happening because you can't sense it. But as we speak now, many of your cells is undergoing the same thing. Okay. So I'm going to stop here because on Wednesday, we're going to continue with the cell cycle. Okay? Any questions so far? Atman. Yes? Atman, you raise your hand. Hello? Question? People still here?
Yes, we have no question.
Okay. Akmal, you raise your hands just now. You have question? Okay. Okay, now, that's it. We have a few minutes left, 2 minutes left. Do you understand what's being taught?
Yes, doctor.
Okay, that's number one. Number two, do you find it easy to understand when I talk? Is the pace correct? Is it too fast? Is it too slow? Or I give too much information when I talk? What do you think?
I think your lectures were fine.
Fine. Okay. Not good, just fine.
Yes, good.
Okay. Anything else that you think I can improve for the lecture?
Doctor, would you upload the lecture notes?
Okay, that's always the question that people keep asking. Lecture notes will be uploaded, but only say, for example, that this week I covered the lectures. At the end of the week, it will be made available. So always after the lectures, it will be made available. That's number one. Number two, some people ask for recording. I will record some of the lectures because we were told that now majority of lectures will be synchronous because we want to move towards face-to-face. I don't know how we're going to move towards face-to-face, maybe early next year. But I will record some of it, especially parts of the lecture which I think is a bit more complex. People might want to revisit that again, and that will also be made available. Okay? Anything else? Is that okay, Afina?
Yeah, thank you.
Is there anything else?
Doctor,
Yes?
is there any attendance we have to fill in?
Yes. But at the moment, I don't take attendance because I don't want to see how many people keep on ending up on my lectures. So far, it's full attendance. Anything else? Okay. Everybody seems to be happy. Okay. So I hope next lecture there will be more interaction. If there's nobody here, I'll start calling names. Okay. If that is the case, I'll see you again on Wednesday. You can use the same link. So I see all of you on Wednesday. Until then, take care. And make sure you have breakfast before my lecture.
Okay, doctor.
Okay.
Thank you, doctor.
Thank you.
Thank you, doctor.
Thank you.
Thank you, doctor.
Thank you, doctor.